Now that we've learned all about the types of mutations and causes of mutations that we might find in a cell, let's talk very briefly about how a cell might repair them. There are several different types of mechanisms that we can use to repair mutations in a cell. We're not going to cover these in a ton of detail. This is not the exclusive list of repair mechanisms, but usually by the end of the semester, as it's winding down, we're starting to run out of time a little bit. So we're just going to talk about a couple of types of repair mechanisms. Our first repair mechanism is called mismatch repair. This happens when the wrong base pair has been inserted into a DNA strand. Now, the problem is that the cell needs to know which one was the original correct sequence, because all it sees initially is that the base pairs don't match. So for example, here we've got a guanine paired with a thymine. So this pair doesn't match, we need to fix it. But we need to know if the T is the original or the G is the original. And the cell recognizes this by methylation. So methylation is the addition of a CH3 or a methyl group to the DNA. Now, this is a physical structure alteration. Sometimes methyl groups are added to our DNA, and we will talk about this in chapter 12. And so once the DNA has been replicated, these methyl groups will be added in as part of a replication of the overall structure of the DNA. So this is an addition that happens to the nucleotides. And immediately after replication, the new strand has not been methylated yet, so it hasn't been fully processed. And so the cell can recognize the original template strand by the fact that it has already been methylated. And so it can bind to, or proofreading mechanism enzyme can come in, bind to the strand, recognize which one was the original strand, and put in the correct base pair. So then we need an exonuclease that is going to pull out the one that we need and insert the new nucleotide. And so we can see that happen here. The cell recognizes that the T is the original because maybe not that particular T, but somewhere in that strand, a methyl group has been added. So we know that it's the older strand or the parent strand. And then our mismatch repair complex comes in, binds to the region that needs to be fixed, replaces by exonuclease, and inserts the correct nucleotide, which in this situation is the A. So now we have correctly fixed our mismatch based on the old template. We mentioned that sometimes base pairs or nucleotides are chemically altered due to a mutagen. In the process of direct repair, the altered nucleotide is fixed in place. So for example, we might have one of those thymine dimers where in our sequence, thymines bind across, our thymines should be binding across to their adenine, but because of UV damage, those thymines bind to one another instead. So during direct repair, an enzyme can come in, break this bond, and restore the bond with the complementary base pairs. Another example is an enzyme called methyltransferase. Sometimes our guanines gain a methyl group. Now this can be due to substances like cigarette smoke. There are certain foods, for example, things that are high in nitrates or even chemotherapy drugs that can force a structural change on our nucleotides. So one example is this 0,6-methylguanine. This means that there is a methyl group that's not supposed to be on our guanine, and an enzyme called methyltransferase comes in, recognizes it, and removes it, and restores the original guanine. So we call this direct repair because it repairs the base that's already there instead of replacing it with a new one. In the process of base excision repair, the cell is attempting to repair small structural changes. So in this case, if we have an altered nucleotide, instead of doing a direct repair, that nucleotide can be removed and replaced. So this would be similar to our mismatch repair, except instead of replacing an incorrectly complemented nucleotide, it is replacing a damaged one. So the base pair is cut out and the correct base pair with the correct chemical structure is put into its place. Sometimes we have slightly larger nucleotide repairs that need to take place. This is going to be a larger than just a couple base pairs, but still a relatively small region within our chromosome. So in nucleotide excision repair, we remove a section of nucleotides. So instead of just removing one incorrectly matched base pair or one chemically altered base pair, we're going to remove a small section. So the same idea here though, the nucleotide is removed and then replaced. So this is specifically nucleotide repairs that are needed because of damage that is distorting the overall structure of our DNA polymerase. And so to do this, we have to separate our strands, cut out the section that is damaged, and then DNA polymerase will come in, fill in the gap, kind of like an Okazaki fragment, and then DNA ligase will seal up that gap behind. The remainder of the slides in this lecture are summary slides. If you would like them, you can write them down, or you can go back to the slides that are available in Brightspace and print them off. And then we also have an example of a genetic disease that is caused by faulty DNA repair. I will just go ahead and let you read through this one. And then our next slide is a list of some other disorders that are caused by an inability of the cell to repair DNA damage.